Hi, everyone. Thank you for being here today. My name is Jessie. I'm a postdoc associate at Darryl Cotton's lab at the Center for Regenerative Medicine at BU. And today I want to share with you some of my work looking at the mechanisms of self-renewal and maturation in iPSC-derived alveolar epithelial type 2 cells. So the alveolar regeneration happens in vivo and in vitro as well in both models. And this normally takes place with a particular set of cells called the type 2 pneumocytes, shown here in green, which mainly acts to self-renewal and differentiate to type 1 cells upon lung injury. And this has been shown in mouse models as early as the 70s in this model of bleomycin injury, where you see labeled alveolar type 2 cells that are proliferating upon injury up to this 12 days post injury and decreases back down to baseline over time. And while this has been shown in a lot of animal models, it's unclear if it occurs in humans as well. What we have generated in the lab is induced pluripotent stem cell derived alveolar type 2 cells through an established protocol for directed differentiation. And these cells proliferate very robustly in culture. And one of the main questions is, why do these cells proliferate so robustly compared to what we see in humans? And also, what are the mechanisms of self-renewal in these iPSC-derived type 2 cells? The main hypothesis of my work is that the ability of these iPSC-derived type 2 cells, or iAEC2s, to self-renew extensively in vitro is due to fluctuating signals that potentially simulate the repair mechanisms that occur after lung injury in vivo. The cells I'm using are from Costas Alexandros in our lab, which have a surfactant protein C TD tomato reporter, so that we can observe and purify the population of iAEC2s over time. And as you can see, these cells proliferate very robustly over time and remain very much surfactant protein C TD tomato positive. And this is just a picture of how the cells look over time when we passage them as single cells, and as they grow bigger, they form these monoepithelial layers of cells here. What I first did was look at how these cells proliferate via EDU staining. So EDU binds to dividing cells. The EDU is shown in the top row over time post-passage, post-single cell passage. The second row is the surfactant protein C TD tomato expression. The third is Hooked. And what you can see is that EDU increases from 1 to 5 days post-passage here in the first row, and decreases in intensity overall. And this is also shown by FACS analysis here in the second plot, showing that there is a dip in the EDU staining, and after another round of single cell passaging, these cells proliferate robustly again. So we're very interested in what drives this robust proliferation at the beginning, followed by the decrease in proliferation. And we chose several time points from this curve to run single cell RNA sequencing. And what we get here is this particular circular structure from the spring plot, which is a force-directed layout analysis of cells, kind of like a UMAP. And as I guide you through the circle, the green shows the 1 day post-passage sample, and as you go counterclockwise, these are the various days post-passage. And the yellow is another 1 day post-passage sample. So the yellow and green plots are overlaid more, which is what we expected for a 1 day post-passage. Now, our cell cycle phase looks similar to what we saw with the EDU analysis, which is that the cells are more proliferative, so in S phase and G2M, or mitosis phase, at the beginning of culture, and then as they grow, they slowly slow down, and then they, at the next 1 day post-passage, they start getting more proliferative again. Now, we're very interested in seeing how maturation signatures change throughout this particular cycle of passaging. And we relied on Killian Hurley's cell stem cell results back in 2020, where he had identified key maturation genes such as surfactants and SLPE. And through this signature, we can see that cells that are most mature in this particular data set are the cells in this spike here, which are around day 5 and day 7 post-passage. And the violin plots are as shown on the left for some of these genes, followed by validation via RT-qPCR. And we wanted to see what was similar or different from this data set of mature cells and proliferative cells compared to other data sets of both human primary alveolar cells as well as mouse cells. So with that, we had used both our own mouse injury model data set as well as a mouse organoid data set from the literature. So for my project, I also have these surfactant protein C Cre-ERT2 mice that are crossed with a rosette toxin A, so that when tamoxifen is given to these mice, type 2 cells are specifically ablated. And this is shown via immunostaining here. As you can see, the prosurfactant protein C on the top row has reduced staining by 1 day post-injury, as well as 2-day post-injury before the resumption of the staining by 2 weeks post-injury, and that is quantified, scored on the plot on the right. We are very much interested in the 2-day post-injury time point, as it seems that that's also the more proliferative, where the more proliferative cells come up. I haven't shown the data here, but I have measured by MKS67 from the bulk lung sample. And I ran single-cell RNA sequencing on this 2-day post-injury time point, and I was able to get 3 separate type 2 cell populations, one of which is DTA positive, which I called the injured population, and one was the proliferative MKI67 positive population, which are separated from the healthy main population. So with these 2 single-cell data sets now, I also combined with both cultured human primary cells as well as cultured mouse cells from the Kobayashi paper to look at the enriched terms from the proliferative sets versus the non-proliferative cells with cell cycle genes regressed out. And what I get is 244 genes that are overlapping between all 4 data sets, both of my iPSC-derived human type 2 cells, my mouse cells, as well as what we have in cultured human type 2 cells and cultured mouse type 2 cells. So what are some of the top genes in this list? I've listed them here on the left. Some of the key ones that I pointed out are FOXM1 and EZH2, which are both transcription factors that are implicated still in cell cycle, but also seem to have other important roles in development and in the body. So I've taken several genes, including these 2 in FOS, and use small molecule inhibitors on our alveolar spheres to see if that modulates either the maturation state or the proliferative state of the cells. And what you see here on the right is an example of this. A vehicle control shown here with inhibitors for EZH2, FOS, and FOXM1 specifically, as well as the surfactant protein C TD tomato channel down below. And what you can see here is that both the EZH2 and the FOXM1 inhibitor seem to reduce the surfactant protein C TD tomato expression. But when we look at spheroid number and spheroid area, it seems as though that the proliferation capacity is still maintained. So in conclusion, I have shown that the IAEC2s in vitro can go through cyclical transcriptional fluctuations. It looks like it's a 2-part model. The first part is from the day 1 through day 7, where there's a proliferation to maturation shift. And what I have not talked about currently is the unknown effects after this maturation peak, just due to the sake of time. And third, I've shown that these proliferative IAEC2s transcriptionally overlap with proliferative mouse AEC2s after injury, as well as overlapping with those from primary human AEC2s in culture. And I got into a little bit of the future directions, which is we want to try to modulate key pathways and transcription factors in these cells in order to activate self-renewal following lung injury, inhibit self-renewal to prevent aberrant growth, and also try to inhibit self-renewal and drive maturation to promote functional repair. So I'm happy to take any questions, and thank you for your time.